I'm pleased to have Dr. Joanna Barbara here with us today. Joanna obtained her Ph.D. in analytical chemistry from the University of Florida. She joined XenoTech in 2007 and currently serves as the Vice President of Scientific Operations. Dr. Barbara also serves as an adjunct graduate professor of pharmaceutical sciences for the College of Pharmacy at the University of Missouri, Kansas City. She has authored or co-authored numerous scientific posters and papers and has represented XenoTech as an invited speaker at various analytical and drug metabolism conferences. Joanna has extensive experience in DMPK, regulatory compliance, and process and project management. As Vice President of Scientific Operations, she is responsible for the development, design, operation, and improvement of XenoTech's scientific division. It is my pleasure to present to you Dr. Joanna Barbara.
Good morning or good afternoon, everybody. We're going to talk today about induction studies. We've been doing induction studies for more than 2 decades now, so we have quite a lot of experience and we've seen changes in design over the years. But in 2017, when the FDA came out with their newest draft guidance, we really saw some more rigor in what was prescribed by the agency. And so that stimulated a lot of questions at the time, and they continue to come. So we decided that we would spend a little bit of time addressing some of them in this webinar today. We'll start out by very briefly talking about enzyme induction basics. I'm really not going to spend much time on that at all, because I'd like to focus on the questions that we get from clients and from competitors and from other colleagues for the majority of the time. Then we'll move into standard study design and what we do with the data once we've got it, and then spend a little bit of time on questions that didn't really fit easily anywhere else, but that we get asked quite often. So the reason that we're talking is because some drugs and herbal substances and some foods even can cause upregulation of proteins. And for us, because we work in the drug-drug interaction space, we're particularly interested in the upregulation of drug-metabolizing enzymes, with the big one that we spend time on being cytochrome P450 enzymes that turn over the majority of drugs that are out on the market today. Generally, this is by activation of gene transcription, and that is a huge sweeping generalization. And often it's by nuclear receptor activation, but that is a whole other topic that I'm not going to get into because that's pretty big and we don't have time. So for us, when we're doing these every day, we're mostly focused on 3 enzymes. We're predominantly focused on 1A2, 2B6, and 3A4 cytochromes P450, and those are regulated by these various receptors, aryl hydrocarbon, constitutive androstenedione, and pregnane X. Throughout the rest of the presentation, I'm going to be saying AHR, CAR, and PXR because those are very long names. PXR activation is the most common. That particular receptor has a very large and flexible binding pocket, so the substrate specificity is pretty broad. And that's a little bit unfortunate in some ways because that one regulates CYP3A4, which is the enzyme most involved out of all 3 in the majority of drugs that are out on the market today, and even still in development as at present. It also is involved in things like PGP and regulation of some other proteins. So it's a pretty important one, and we spend an awful lot of our time talking about that particular activation pathway. Something that's important to note, the reason that this is hugely important from an in vitro perspective is that species differences in this arena are huge, and animal models are very poor predictors of human induction potential. So when these proteins are overexpressed, that really impacts metabolic clearance of various comeds or sometimes endogenous substances. If the drug of interest is inducing the enzyme that is responsible for its own metabolism, then usually that means that that's going to change the clearance of itself, and that's called autoinduction, which is a very interesting topic in and of itself, but it's more common that it's going to induce a metabolizing enzyme that's responsible for other things. So concomitantly administered drugs can then have some kind of drug-drug interaction, and so early on in drug development we need to have a look at drug-drug interaction potential and see whether this is a dangerous thing or something to be concerned about. It usually doesn't rule out continuation of a lead compound in and of itself. On its own, it usually just means that you're going to be doing some follow-up work, and I'll talk more about that later. But it is important because you can really see changes to efficacy of narrow therapeutic index drugs. The common one that people talk about a lot in this arena is the anti-epileptics, carbamazepine, phenobarbital, and a few more. induce P450 enzymes, which speeds up clearance of various comeds, with warfarin being the most common anticoagulant one that a lot of people are on. And because we have more enzyme, then we're turning over the drug much more quickly, which reduces the exposure that the patients experience, and so often you'll see a lower therapeutic effect. So these are the most recent FDA and EMA guidance requirements. The PMDA is actually very similar. While the FDA guidance really brought out some big changes, not so much in what we were doing, but in terms of making the things standard that we did for some clients, but not all. It was actually very refreshing because there was more correlation between the FDA and the EMA guidances. And as a CRO, that's really helpful for us because it can be very difficult if they have conflicting requirements. It can be very difficult to generate standard studies that meet everybody's needs. So these are the very high level points of import that are discussed in the FDA and the EMA guidance. The test system is cryopreserved or fresh human hepatocytes, liver cells. Other systems are considered complementary by the EMA. There's a little bit more to it with the FDA, and I'm going to specifically talk to that in a little while. But both of them say a minimum of 3 donors so that we can explore inter-individual variability. Both of them are emphasizing the 1A2, 2B6, and 3A4 enzymes, which we all expected. There is a slight difference in TA concentrations, so test article or drug concentrations that need evaluation. The intent of both guidances is to look at safety factors and to allow safety factors for those changes in people. But what we will see is that the FDA is really looking for about a 10x Cmax steady state, Cmax unbound steady state to be covered, or intestinal drug concentration. So that's looking at dose in 250 mils usually. Whereas the EMA is looking at a 50 Cmax steady state unbound. So what we typically do is strive for the 50 times Cmax because that way it covers everything. But that can be pretty challenging for drugs which traditionally don't like to be solubilized. These kinds of studies are super important in terms of the positive controls that you use because the whole way that you use the data is a relative thought. So does this induce enough to have a clinical effect potentially is the question. Because lots of things induce, but it doesn't necessarily mean that there will be a problem at the end. And what you need to do is put them in context with known drugs. So the positive controls are very important. And the FDA and EMA agree in terms of suggestions for 2 of the enzymes. So for 1A2, we look at omeprazole, and for 3A4, we're going to look at rifampin. The challenge is with 2B6, where the FDA recommends phenobarbital and the EMA recommends Citco. And it can be very difficult to know what to do in that scenario. So I'm going to talk to that a little bit because that's a question we get quite often. And intriguingly, the FDA now recommends negative control. The EMA still does not, but there's been considerable debate in the scientific community about what the negative control brings over the vehicle control. All right, so a standard study looks something like this. We have what we refer to as supporting or preliminary test. We can't say just preliminary because some of them happen concurrently with the main induction test. I'll elaborate presently. We use cryopreserved cultured hepatocytes. I already mentioned 3 individual donors minimum. The FDA guidance now literally calls out for N of 3. And for us, the cryopreserved hepatocytes are pre-characterized for inductions. We do a 3-day treatment generally at 7 concentrations. That's our standard. And we very often get the question, if I do fewer concentrations, can it save a little bit of money? And it's really, in my mind, it's a very false economy. It's going to save you a tiny amount of money, but the fact is you're still doing all the work and you're still doing the cell culture, which is a giant piece of the price. And if you end up with a positive result, now you're not going to be able to define your induction curves. So what actually happens is, yes, you spent a little bit of money, but you don't get the endpoints and the data that you need. It's not worth it. So my advice is don't skimp on that. We do have to measure the concentration in the cell culture medium now for absolutes. We didn't have to previously. I am going to spend some more time on that because that's an area we get a lot of questions. It usually means there's going to be an LC-MS method developed for that drug in that medium. And basically, you're going to end up with a table of concentrations at the end saying this is what the cells actually saw over the incubation time course. We basically always do quantitative mRNA measurement. mRNA is the required endpoint by the EMA and is a recommended endpoint according to the FDA. It's also a little bit cheaper and easier to achieve than if you have to do all of activity measurements. We do activity by LC-MS where applicable. So sometimes you need both. Sometimes maybe people want to explore the activity versus the mRNA, but sometimes you have to do activity, and I'll explain when and why later on a few slides down. What we're looking to do is achieve, does the drug cause a change in the enzyme expression over your vehicle control? So we're looking to evaluate fold change numbers. It's a very simple endpoint. And then if we see a positive induction response, our standard approach is to go ahead and calculate EC50 and Emaxes. That's standard. We will talk quite a lot about those presently. The supporting and preliminary assays are things that we've been doing for a long time, but they weren't always standard previously. One of the ways that we do this is we kind of have to schedule and time out how the study happens. And so there are two supporting experiments that we have to do prior to the real induction assay, because those are going to dictate the concentrations that we can truly test. We're targeting 50 times Cmax, or the high dose, but that doesn't mean we're going to be able to do that from a practical perspective. So the first thing that we do is solubility testing, because this is a really high concentration assay. Sometimes we're talking about high millimolar quantities depending on what kind of dose and exposure people are expecting. So we start out with the simple stock solvent solubility. We have to be able to get high enough stock solvents to keep the percentage of organic very, very low in the assay, because you know what? Biological tissues do not appreciate strong organic solvents. We also then have to move into the more challenging piece, which is test system solubility. Quite often, clients will say, well, I have solubility data. I know how soluble this is. Why are you doing this solubility testing? And unfortunately, the answer is hardly anybody uses the cell culture media that we use in their normal solubility testing, because there would be no reason. We use very specific things to support this very specific assay. So I have yet to see a client that comes to us with solubility data at the concentrations we need in the medium that we need. So it is important that we do that, because at that point we're able to find out exactly what kind of concentration range we really truly can evaluate. And the FDA says the limit of solubility is acceptable, so we know that that's a good way to approach it. We also do cytotoxicity testing prior to the experiment itself. We're doing cell morphology observations to look visually at cell health. So that's just taking pictures with a microscope of the cells and seeing if they still look the right shape and have appropriate nuclei and things. We're doing that all the way through the assays anyway, and we always do. But now we more commonly will do a lactate dehydrogenase release assay early on to find out if all the cells will be poorly impacted by the high concentrations that we're looking at. When cell membranes break down, lactate dehydrogenase is released. And so typically we'll do a little experiment early on to find out at what point the cells can no longer tolerate the drug. And that's the other limiting factor for the concentration ranges that we test. Then the third supporting piece is what we call spent media analysis, because to us, it's the used up cell culture media. Sometimes this gets confusing for people who don't use the same words as we do. You know, every company has their little traditions of how they refer to things. So what all that means is we're analyzing for the concentration remaining in that medium sample after whatever incubation time. And so we're going to do that on the last day of treatment all the way out for 24 hours. We often get the question, why? What is it for? Well, the brief and not very helpful answer is it's for the FDA who specifically requested. But the more useful scientific answer is twofold. One, it's important to know what kind of concentration is actually being experienced. There may be a need on the back end to correct the data for that concentration if you're going to calculate EC50 and Emax. You know, maybe you put in 100 micromolar, but actually 50% of it got lost immediately to non-specific binding, at which point you've only got 50 micromolar, and your EC50 is actually a very different value than it appears to be using the theoretical concentrations. The other reason that it can be useful is if the drug is cleared very quickly in the hepatocyte cultures, you might need to dose more than once a day. That is specifically discussed in some of the guidance documents, and basically it says you have to justify why you're dosing more than once a day. So it's extremely rare. I personally have never seen a study. I've been here 12 years. I've never seen a study where we have had to dose more than once a day, but I'm informed that it has happened very rarely. I think it was once in 17 years, if memory serves. But theoretically, that is one other use for this particular supporting assay. So let's talk about cultured hepatocytes a little bit. We use cryopreserved versus fresh, basically since 2012, the last guidance that came out. That's been hugely helpful for scheduling, because back in the day, it used to be that you would have to wait for fresh liver tissue before you could even run the assay. So if you're trying to do 3 cultures, that can take a long time. It also helps because we have characterization going in. We know whether this lot is going to be a useful lot or not, and we get consistent data all the way through until we run out of this individual lot. These are some pretty healthy cells shown in this top right. That's what we're looking for. That's what we want to see. You can clearly see cells. Cell culture is not my forte. We have a lot of people who are very good at it. So what I typically look for is, are there holes in there? If there are holes in that picture, something's wrong. This one doesn't have very many holes, so we're happy with this. When we make hepatocytes, because of course we sell hepatocytes here, so we are very fortunate, we have access to whatever hepatocytes we want. When we make them and we find out that a lot is going to be attaching and going to be a nice useful lot, it is our standard practice to move forward and characterize for induction potential. So our data sheet for a lot is going to come with something like this. This is a picture of a piece of a data sheet. Down here is the induction data. This one is an older one, so it doesn't have any 2C data. But it does have 1A2, 2B6, and 3A4 data, and you can see that we have the mRNA fold induction response from omeprazole, phenobarbital, rifampin, and then we have the enzyme activity induction response. The cutoff for mRNA fold induction is 6, so all of these are above 6, so we're happy with those. The cutoff for enzymatic activity induction is 2-fold, and so this is a nice slot that we would go ahead and use internally, and we would recommend for use externally. That's our standard approach. We also have some lots that are characterized for relative induction score, about which I will discuss more in a few minutes. And so very occasionally we get the answer, do we have to use RIS, relative induction score, characterized lots? The short answer is no, and the majority of our clients do not. The longer answer is it depends, and it depends what your particular strategy is for using the data on the back end. If people need them, we have them. If they want to use our data, we have them, but the vast majority of clients do not use those. So something that's very interesting to me is, why not immortalize cell lines? So I'm a scientist by training, and I love science, but I also now am responsible for operations. So I like things that mean that you can schedule all the time and you don't have to characterize again and again and again, because they're just more practical and easier to keep moving. So when we saw that the FDA guidance now suggests that you could use immortalized cell lines for real induction data, we did, of course, look at it again, because it raises the question, should you? And we do use some immortalized cell lines for a couple of our other pieces of work. It's still complementary per the EMA, so people doing EMA submission would not be able to use it. But the more we looked into it, the more we recognized that actually all the old problems are still there. They haven't gone away. Most of these come from liver tumors, not all, but most, and they basically are not hepatocytes. That's the big problem with them. They each suffer from various low expression of certain things, and so you can use HepRRG for a 3A4 induction study, but you're not going to be using it for a 2B6. FA2N4 and HepG2 have hardly any basal CYP enzyme expression, and so they don't respond well to 3A4 inducers, thus rendering them not all that useful. HepG2 is useful for 1A2 because it has very high AHR expression, but as Chu put it in the perspective paper from actually 10 years ago, basically no one cell line affords an exact reproduction of a hepatocyte, and so they're not very good for a replacement for human hepatocytes. So I guess the way to look at it is, we're looking to see what would happen in a hepatocyte. Why wouldn't we use a hepatocyte? So that's a place that we didn't really go for induction, but we do have the conversation every now and again. I mentioned earlier that it's very important that you use the right positive control inducers. And so here are the agency recommendations. The FDA and the EMA both recommend omeprazole. FDA also has lansoprazole for 1A2. The challenge is, as I mentioned before, the phenobarbital versus Sitco for CAR, and then rifampin for the PXR enzymes. So why did we go ahead with phenobarbital? The cynical people in the audience would say because most of your clients are going to be US based because you're in the US. And that's a fair argument, but it's not the reason. We selected phenobarbital because it is more predictable and useful. We can do Sitco, we could if we wanted, but we stick with phenobarb most of the time. It is true that Sitco is selective for the CAR pathway, which is why it's recommended by the EMA. The problem is the fold change response is extremely variable, whereas with phenobarbital, you can traditionally get results where the induction is greater than six-fold for mRNA. That is not true of Sitco. Sometimes it won't even be a good 2. So it's not a very practical choice for us. But also, in people, there is considerable crosstalk between the CAR and the PXR pathways. So one of the arguments for Sitco is that you've eradicated that crosstalk, but of course that's not realistic because in real life you're going to have crosstalk in a person. So an argument for phenobarbital that sometimes is put out in the literature is that actually you want to account for both because that's going to be a more complete test system. So that's why we use phenobarbital. Our incubation is based on sandwich cultured hepatocytes with a collagen overlay. And we typically will do about a 1-day adaptation, 3 days of treatment at multiple concentrations. We talked about the need for as many as we can and get, preferably 7. You have diminishing returns when you get too high. And we're going to treat them with a vehicle control, increased concentration of drugs, then our negative control and the positive control. So that's where the omeprazole, the phenobarb, is going to be. Then we're going to collect samples out to 24 hours post the final treatment and collect the spent media, that's that cell culture media from the dosing. If we need to do enzyme activity, that's when we'll do it, and then we'll collect cells for mRNA harvest. Then we send off the samples for RT-PCR or LCM-SMS, depending on whether we're looking at activity, drug concentration, or mRNA expression. And then an awful lot of activity happens before we get to the next step, but I'm going to skip it all because we're going to look at data. So this is what I consider first pass data. It's a little bit of a misnomer because there's already quite a lot of analytical data processing that happens before we get here. But the curves, the pictures that people want are here. So this is representation of the fold change data over vehicle control. And it's the exact same order I just said, where we have vehicle control, increasing drug, negative control, positive control. This one is a beautiful positive response. It shows gorgeous concentration dependence, and we don't see any signs of cytotox at the end. We're actually greater than the fold change response for the positive control, and the requirements for a positive are greater than a 2-fold change over the vehicle control and greater than or equal to 20% of the positive control response. So this is an absolute definite positive with no questions. This one is what a negative response looks like, and we have to, because it's so negative, we have to blow it up. So basically, all of the drug concentrations are floating around the vehicle control, which is setting us at 1. But the negative control did what it's supposed to do, nothing happened, and the positive control did exactly what it's supposed to do. It induced beautifully, so we're very confident that the induction pathway was working, and this is a true negative. When it looks like this is when it gets a little bit tougher. So it sort of looks like there's induction, but it's not a pretty concentration-dependent response, and also we're seeing a little drop-off here. Usually if that's related to cytotox, you're going to see that not just in one of the bar graphs. You're going to see that across a couple of enzymes. Not always, but usually. So in this case, it's tougher to confidently say, yes, it's beautiful concentration-dependent induction. It still technically is a positive. And so it still warrants consideration, because if you look, we're greater than 2-fold at both of these concentrations, and we're greater than 20% of the positive control response, which would be, I don't know, call it there. So it is a positive, but it's not as straightforward to deal with as the first one. For those positive responses, we're going to go ahead and we're going to calculate the EC50 and Emax. It's just like calculating kinetic constants for an enzyme reaction. It's basically the same principle. This is tricky though, because you have increasing sigmoidal curves, and so it takes some finesse to really be good at fitting them. So what we're trying to do is figure out what concentration would be associated with half maximal induction and what the maximum fold induction in vitro is, because those are comparators that we can use to assess whether this is risky for patients or not. These are two graphs. They show the same data set, but this one is fitted with the sigmoid 3-parameter equation, and this one is fitted with the Hill 3-parameter equation. In both cases, we get pretty similar results. So the Emax is where we max out induction. That's pretty straightforward. Then at 50% of that, the concentration associated with that is our EC50. And the main difference is that the Hill 3 forces this to be the maximum induction response. So we typically will use the sigmoid 3 where we can. But we calculate those for all of our positives because at the end of the day, knowing it's a positive isn't very helpful unless you know what to do with that information. So what does it mean? We talked about what a positive is. It is very important to recognize that even if this happens for one enzyme, one donor, that is still a positive result. We're pretty excited because the FDA and EMA agree now, so that makes our lives a little bit easier. But we do get the question, well, why is that a positive? And the answer is that both agencies are taking quite a conservative approach to patient safety, which is absolutely understandable because that is one of their main missions. We know there's going to be inter-individual variability, so we want to know what the worst case scenario is. And the worst case scenario is whatever happened in that one donor in this particular case. So what do we do about it when we have a positive response? Every now and again, someone will say, is my compound dead then? That's a pretty rare question, but it does come up, so I'm going to address it. There is no drug that has been either withdrawn or prevented from moving forward solely because it was an enzyme inducer. There can be complications that mean that that's a contributing factor, but just because you see a positive induction response in no way means that the drug is finished. There's so much more to do before you know what that means. If it is a CYP3A4 positive response, then you have to move forward at some point and evaluate CYP2Cs because they're also upregulated by the PXR pathway. Doesn't necessarily have to be in vitro, but it will need exploring at some point. So the next thing that you're going to want to do once you've established that and generated all the data you need is to look at mathematical models and see what that actually means in context. We'll talk a lot about that in just a few minutes. There is a very nice IQ induction working group paper that came out in 2018 that talks about statistical power basically in this context and recommends doing a fourth culture to try say, well, it's only 1 out of 4, so it's not as problematic as it appears. The arguments are very, very good. It's worth reading if you have the time. The challenge for us as a CRO is that you're still going to have that one positive, and to the letter of the guidance, you're probably still going to have to do something. Now, if it so happens that you have statisticians on staff that can go and make that argument about what that really looks like, then that's great, and it's absolutely plausible to go and have a conversation with the agencies. Most of our clients don't have that ability, so we typically take a more conservative approach than that. So CYP2C enzymes are induced through the PXR pathway too. So we are going to need to explore them if we have positive 3A4 responses. The question we get an awful lot is mRNA versus activity for CYP2Cs. It is absolutely known across industry that the mRNA fold change for CYP2Cs is very variable and often doesn't even make it greater than 2. It's a real problem. So we recommend activity for the 2Cs. It's a much more reliable endpoint. It is possible that the fold change may still be less than 2, but it's much less common, whereas it happens with mRNA all the time. There are a lot of reasons for that. Proposed in a different induction working group paper from 2017, which is also worth a good read. But I'm not going to evaluate those here. The reason being, I wish I knew what the true reason was, because if I knew, then we could solve that problem and we would win all the induction work forever. But we don't know. So it would just be discussing options. So then the next logical question is, well, why not look at the characterization data for the lots that you have, pick the ones that induce the 2Cs the best, and use those, and then you would get a reliable mRNA fold change, right? Well, no, because that's exactly what we do. We scour the data, we go, those look like they have the potential to be the best for 2Cs, so we're going to use those, but it's just not predictable. These are cultured cells. They're not going to be identical every time you thaw them and culture them. It just doesn't work that way. So what we really need to do once we have these positive responses is start thinking through modeling, essentially. There are a lot of mathematical tools for risk assessment, and basically the FDA guidance gives you a nice little pathway to follow for how you deal with a positive response. So that's what we propose too. And you can use correlation methods, basic and static mechanistic models, or get into the really complex area of physiologically based pharmacokinetics, PBPK modeling. And so what we typically do is start with the simple and move to the complex when we need to. In order to do these calculations, we usually need dose and Cmax data, or at least projections. Just in and of itself, the fact that there is no Cmax does not necessarily mean that we can't do anything to look at risk assessment, but it's a more custom situation. That means you need to talk to us before we know what to do next. So the first one in the guidance is correlation methods. This is where that relative induction score comes into play. And the guidance basically says, calculate these things, plot them. It's essentially calibration. We have a calibration curve here. This is a risk curve for one of our lots. It's always in the context of midazolam AUC changes in the clinic, because that's how we scale these things. And so we have a bunch of known compounds that we calculated the relative induction score for. This is relative induction score for low risk compounds. This is relative induction score for higher risk compounds. Rifampin, that we all know is clinically relevant, is in here. And then you plot your drug on this curve and you go, well, you know, it's down here, so it's not too much of a problem, or it's up here at this high risk, so I really need to be worried about it and do something a little bit more complex. You can also use the slightly simpler approach of Imax over EC50. So this Imax U is Cmax unbound, is maximal unbound plasma concentration of the drug. I always like to point that out because we call it so many different things and sometimes it gets confusing. So this is a useful way to put the induction response that you're seeing in vitro into context. Some people do it with our data, and that's when they want to use the risk characterized lots, but more people are going to do things with their own data or use some of the other tools. That's the more common approach for our clients. The EMA also allows the use of AUC over F2, which is very useful in situations where you can't really define an Emax or an EC50, which does happen. Sometimes those curves are basically asymptotic. So in that case, you can use F2, which is the theoretical concentration of the compound that would end up with a twofold increase of baseline. So that's something that you can use per EMA, sorry. If that's not the preference, then the other thing that we do, and we actually do pretty commonly now, is calculate an R3 value. This is the predicted ratio of the area under the curve, or the exposure change of the drug in the presence and absence of an inducer. It's a pretty simple equation. Again, you do need Emax and EC50, which is why we calculate those. And basically, the only challenge here is the scaling factor, which sometimes you know, but more often you don't, and you have to set it to 1. There is a cutoff value for the FDA now of if the R3 is less than or equal to 0.8, then a little bit more work needs to be done. Again, that doesn't mean you're going immediately to the clinic, but it does mean that you're not done with this question yet and you have to keep working on it. So I always say we need to look at it more. If you do need to look at it more, you can go to the more complex model, which is the static mechanistic model, which looks a bit intimidating at first until you actually try and do it, and then it's really not, especially if you're doing it by hand, which is way easier for me than spreadsheets, but is not the way that we typically do it for clients because we like to do things fast. It's very interesting in that we used to use this and do net effect, so we would include the effects of induction and inhibition and see what that meant overall for a patient. And now we don't. Now the guidance specifically says, no, you're just going to look at the individual effects. So if you look at the equation, we have this guy is about direct or reversible inhibition. This one is to do with time-dependent inhibition. And then this one is induction. So we used to fill out all of these and come up with an overall AUCR. Nowadays, when we're looking at induction, we're going to set all of these to 1, which is 1, so that we can look at the absolute worst case scenario for an inducer that doesn't have a mitigation effect from an inhibitor. And that is the reason why the net effect model isn't recommended by the FDA anymore, because they're trying to be super conservative. They want to know what is the worst that can happen if you had an inhibition effect? What is the worst that can happen if you have an induction effect? So basically, you're going to calculate your comparators here based on the gut or based on the liver, depending on which one you're doing, and calculate out your AUCR just for the individual effect. Now, if that's still less than 0.8, now you've really got to do a little bit more heavy lifting. And that takes you to usually mechanistic modeling or a lot of people will at that point go straight to the clinic and do some drug-drug interaction studies. The PBPK is a good idea even if the ultimate endpoint is going to the clinic, because it really helps you design an appropriate study, helps you with things like exclusion criteria. The other side of this is if you really need to, you can always just use exclusion criteria and focus on that and labeling. But the majority of people don't want to do that. So there's a lot of guidance out there. This isn't something that we do here. But the DDI guidance that we've been relying pretty heavily on for most of this talk provides a pretty nice framework with how to think about approaching PBPK. And then once that's been digested, there is actually a guidance specifically for that that is much more prescriptive and says you need to address this, you need to address this. And so if that's a route you're going, but you're not really very well versed in that area, I strongly recommend taking a look at those and using those to help you make the right decisions. So there are a few questions that we get all the time that didn't really fit very well into our normal workflow. The most common one is, when should I place the study? And unfortunately, the answer to that is it depends. It depends on your program, it depends on your risk tolerance, it depends on your indication. But conservatively, you're going to want to know induction potential before you get into a patient. So what we're seeing is that people are actually doing this earlier and earlier. We're doing quite a lot of definitive induction work pre-IND at this point. Not always, but a lot more than we used to. So my advice is at least get some data as early as possible, even if it's just a 3A4 screen, because you're going to want to be planning for that as you budget and timeline out your program. Another question we get quite often is, do I need a negative control? It is absolutely fair to say that it may not add a lot of value. It's standard for us anyway, and it doesn't impact cost in any way. It's not going to be less if we take it out. In fact, it might be more because we'd have to rework our plate layout. But it really doesn't make much difference. We've always done that for standard for a very long time now. There's no downside to doing it. But in short, the FDA says you need one, so it's probably a good idea to include it. It's pretty trivial inclusion. And then the third one is, can I use my validated BioA method for the concentration measurements in the spent media analysis? We get this question very, very often. And unfortunately, the answer to that is no, we can't. And the reason is, usually a regulated BioA method is going to be for pretty low concentrations because we're talking about plasma exposure, and it's in the matrix plasma or some extract of plasma. Well, a method developed for that is very unlikely to also work for a high concentration range assay in an extremely different matrix. That's why in BioA, if you're doing a rat study or a dog study, you have to revalidate in the rat compared to the dog, because matrices are very different. Instrumental responses are very different. The curves may not behave at all the same. So unfortunately, we can't do that. We do like to receive method information early on because it can speed things up. It can narrow the timeline for everybody because we know where to start with columns and things. But unfortunately, they're not going to directly translate. And then there are a couple of more complex questions that we need to address. Sometimes in these studies, you see downregulation of the enzymes. And first of all, that does not necessarily mean that that's a thing that will happen in the clinic. There is, I mentioned earlier, this 2017 Induction Working Group paper. It's very interesting. It talks about challenges with 2Cs and it talks about downregulation. Now, there is a cutoff for what does downregulation actually mean. When do we really truly have downregulation? And the EMA says 50% decrease, and it should be concentration dependent. Usually when you see that, it's a cytotoxicity effect and it's not actually downregulation, but sometimes you can rule that out. So the first thing that needs to happen if you do see this is this just because of your mechanism of action in some way? Because if it is, it's a very different consideration than if it's nothing to do with that. Is it really just cytotoxicity looking a bit weird? Because quite often, most of the time, I would say that's the true reason. Or if you're looking at activity, is it that you also have enzyme inhibition? And so that's one of the reasons that you need to know at some point, not necessarily in the same study, whether or not you have enzyme inhibition. If you've ruled out all of those things, the industry thought is that you may need to do some mechanistic studies to evaluate the cause. We do not do those. But there is literature out there, and it's referenced in this Induction Working Group paper, that you can look at to see what that means. When I look at that and I think about that, That sounds like a very big, time-consuming undertaking, almost like a needle in a haystack situation. And unfortunately, there isn't a lot of useful research in this data to do a really good job of that. So it's not a trivial consideration. And then finally, we also get asked a lot of the time to look at UGTs and transporters. We hardly ever actually end up doing the studies. UGT we will do sometimes, transporters are very rare. And we absolutely can look at that. I mean, if we can get a primer for a thing, then we can measure mRNA for that thing. The question is, how useful is it? UGT induction in people is usually associated with much lower magnitude drug-drug interactions than CYP induction. So that's why it's not focused on as heavily. But the real reason that we typically don't do it is because it's very unpredictable in vitro. And again, this is an industry issue. This isn't just us. Very often you won't see fold increases greater than 2 or at appropriate levels that you need to be able to use the data for decision making for UGTs. Here we have some data. The top is the activity data and the bottom is the mRNA data for a few of the common UGTs with some common inducers. And you can see that we only make it over 2-fold in one of the situations by activity. So it's very difficult to get a useful, reliable response in this scenario. The mRNA is a little bit better. We did a little bit better in 2 scenarios. We were able to get useful data, but it's still pretty low. And it's also unpredictable and very variable. So we absolutely can do it, but we're not going to be guaranteeing positive control responses because it's just not possible. The science just isn't there right now. And PGP is the most common transporter induction question that we get. We can and have done it. We don't typically recommend it. And it actually calls this out in the FDA guidance, where basically it says that the in vitro methods to evaluate induction of PGP and transporters aren't very well established. There isn't enough data to know how useful it is. And so at present, the FDA does not currently recommend in vitro evaluation of transporter induction. And that is also our stance, because it's just not something that you can contextualize once you've done it. So we talked a bit about study designs from a scientific and a regulatory perspective. It's very important that whatever study you do is going to give you the endpoints that you need so that you can make decisions. And so sometimes, you could get a smaller scale study, but it doesn't really help you because at the end of the day, A, you can't submit it to the regulatory agencies, or B, it didn't answer the questions and help you know what to do next. A positive result, even from one donor, is a true positive. But it doesn't necessarily mean that you're in trouble. It just means that there needs to be some form of appropriate follow-up. Unfortunately, we're talking about science, so things like the 2Cs and the UGTs are not well defined yet, and some aspects are not perfect. That's just the way it is. And we have to make decisions pretty carefully based on the program needs. Also, you can tell from the stuff that we've talked about today that when we take client questions pretty seriously. I am in the fortunate position of having 130 colleagues basically in this office, and so we can talk at length when we need to. I do need to acknowledge some people. Becky Campbell, who is going to be my partner in crime when we get to Q&A, who basically taught me everything about induction because she's our induction expert, and Madison Knapp, who helped with material, and Dr. Brian Ogilvie, who teaches me things every day.
Thank you, Joanna. And while we wait for any additional questions, I want to mention that information about our services, papers, posters, other webinars, and more is available on our website. If you would like to be notified of future webinars, you can submit a webinar topic request form or register for our newsletter. If you have any further questions regarding Joanna's presentation or any of XenoTech's contract research services or products, please feel free to ask them in the post-webinar survey or contact us through our website. Okay, this wraps up today's session. Thanks to all of you for taking time out of your day to join us, and thank you to Dr. Barbara for her wonderful presentation, and to Rebecca for participating in the Q&A. Have a great rest of your day.